Hello again. You'll see that I'll be moving fairly quickly in the next few sections because I've already explained them in the previous videos. This part talks about the Human Genome Project. The Human Genome Project will be covered in greater detail in Chapter 9. You can also have a look at my YouTube channel. There are some videos on Human Genome Project. I think it's labeled HGP, and they refer to certain documents that I've also put on Spectrum. It talks about the goals and the strategy and things like that. But to briefly summarize it, it says the Human Genome Project, when it was established very much early in 1990s, it has three different goals, which is one, to develop mapping and sequencing technologies, right? And then because at that time they can only sequence about effectively about 700 bases, and to sequence a large fragment it was very expensive, right? Very uneconomical. So what they want is to develop sequencing technology so that they can reduce the cost of sequencing. Secondly, in order to get a good genome reference, they thought that you will not be only sequencing the human genome. You also need to sequence other other reference genomes so you can do comparison. In this example, it says 5 model organisms, I think include Arabidopsis, what was it, Drosophila, things like that. So they need these different organisms so that you can compare, and then also to investigate the ethical and legal and societal implications once you have this information about the human genome. Actually, there are several other goals. For example, establishing databases not only to store information but to allow sharing of information globally. There's no point in having sequences but nobody can have a look at them. And the other one is also to develop bioinformatic tools that allows you to analyze the genome, and not just because as much as mentioned much earlier, getting the sequence is the easy part. Understanding them is the bigger challenge. I'll move on quickly. Then it talks about the first human genetic maps. I already mentioned you what are maps. Maps is having markers on landmarks on the human chromosomes. It says one of the first maps is the RFLP, restriction fragment length polymorphism, which you get depending on the absence or the presence or absence of cutting sites. That's different between different individuals. And then so you get a map that's just one marker per 9 million bases, which is not a very high resolution. Then they moved on to microsatellite markers. If you're not familiar or you don't understand what microsatellites are, this it's actually a repetitive sequence, and they are very variable. Why they vary? Because when replication occurs on the regions that has these microsatellites, mistakes do occur. But you're not talking about mistakes that occur every day and so on. You're talking about evolution over over thousands and hundreds and thousands of years. But there is an evolution. So but these are the microsatellites, and then I think the current map is actually based on microsatellites. Although microsatellites are very variable, the flanking region, the regions flanking the microsatellites are unique, and this flanking region is the marker that allows the recognition of its position in the human genome. It's unique, right? So establishing high density DNA marker maps and clone contigs. Again, this I think I've mentioned all that. So hopefully when you read this title, you'll know what it means. Establishing high density DNA marker maps and clone contigs for each human chromosome. So this is what they do in the early parts of the human genome. So you have established markers, established landmarks, and they want to have references that are dense enough, so that dense enough meaning 1 every 20 kb or and so on, so that it's easy to refer to. And in this section also, it talks about STS sequence tag sites and also ESTs express sequence tags. Okay. Now this one requires a little bit of explanation. I will talk about this in the next video. All right. See you later.